Okay, I think we can go ahead and get started. So thank you all for coming to the December StaPH-B call. Today we have Li Zeng with us from the Riverside County Health Public Laboratory in California, and she'll be presenting on extensively drug-resistant shigellosis in Riverside County. Take it away.
Hi everyone. So I start right now. Do I start right now? Okay, let me
Yep.
share my screen first. Can you see my screen?
Looks good.
Okay. Hi, everyone. My name is Li Zeng, a bioinformatician from Riverside County Public Health Lab in California. Today I will present my research on genomic epidemiology of extensive drug-resistant Shigella, or you can call it as XDR Shigella, in Riverside County. The presentation will focus on transmission patterns and possible cross-county spread of XDR Shigella within Southern California. To begin, here is a quick overview of Shigella infection, or can call it shigellosis. Shigella causes acute gastrointestinal illness, including the diarrhea, vomiting, stomach pain and cramps, and also fever. The infection dose of Shigella is very low, normally like as few as 10 to 100 organisms can cause the infection. And Shigella spreads easily through fecal oral transmission, contaminated food, water, or format, and person-to-person contact or sexual contact. Yearly, Shigella infects estimate of 450,000 persons in the United States and with more than 6,000 people hospitalization. There are four major subgroups of Shigella. Today I will focus on the two most common subtype of Shigella called Shigella sonnei and Shigella flexneri. The CDC defined the XDR Shigella as strains resistant to all commonly recommended empiric and alternative antibiotics. Nationally, we've seen increasing XDR Shigella infections in specific populations, including men who have sex with men, persons experience homelessness, immunocompromised individuals, internal travelers, and people living with HIV. Why I'm interested about the XDR Shigella in California, in Riverside County and Southern California, because nationally we've seen increasing XDR Shigella infections in specific populations, including men who have sex with men. California identified a steady increase of antimicrobial resistance among Shigella isolates from 2018 to 2024. Most importantly, the proportion of isolates resistant to all the five antibiotics has continued to rise. We can see from 2022 to 2024, almost 15% of the Shigella isolates have been identified as resistant to all of those five antibiotics. So this rising trend raises concerns for public health response and emphasizes the need for genomic surveillance of this XDR Shigella. Traditionally, Riverside County's Shigella specimens were sent to the state lab for further testing. The state lab normally they sequence approximately about 1/3 of the submitted specimens. In total, 27 of XDR Shigella isolates from Riverside County were sequenced from 2020 to early 2024. But I have to tell you, like majority of the cases 17 out of 27 were reported between late 2023 to early 2024. That means the XDR Shigella rates are increasing recently. Specifically, most cases in Riverside occurred among men living in areas with large men sex with men population. This is a group identified by the CDC as being at higher risk for XDR Shigella infection. Besides Riverside County's cases, to better understand regional transmission, we further expanded our analysis to include all 149 XDR Shigella whole genome sequences from the 5 counties in the Southern California metropolitan region. For confidential here, I just use the Riverside County's name and use letters to represent other counties' name. As shown here, the County A has contributed the largest number of XDR Shigella isolates, followed by Riverside. And the County D, its first whole genome sequenced XDR Shigella cases only in early 2024 and hasn't been reported before 2024. So all of those combined data allow us to examine potential transmission patterns and genetic links across county boundaries. So there are 2 major questions I want to answer for this research. First is, what are the transmission patterns of XDR Shigella in Riverside County? The second is, are the 4 newly identified cases in neighboring County D directly linked to cases in Riverside County? Because geometrically, County D is the neighbor county of Riverside. We obtained all the 149 genomes from NCBI and performed the whole genome SNP-based phylogenetic analysis. On the left, here are the phylogenetic trees from the Riverside County cases only and all the cases in Southern California. On the left, you can see the tree of Riverside County isolates. It can be phylogenetically. We identified the 3 major groups. I marked them as different colors. So the group 1 is Shigella flexneri cases, and group 2 and group 3 both are Shigella sonnei. The same pattern has been identified in the whole Southern California cases. Also, it has been separated as group 1 to 3. And the group 1 is Shigella flexneri, and group 2 and 3 are Shigella sonnei. Each group shows distinct clustering patterns and transmission signatures. I will explain them in detail. So first, let's look at the phylogenetic tree of XDR Shigella isolates from Riverside County only. Here, different colors indicate the year in which each case was reported. The purple is the early case 2020 to 21. The green is 2022. The light orange 2023 to 2024. First, the group 1 is Shigella flexneri. Most of these cases occurred in 2022, with only 1 case reported in 2023 and 1 case reported in 2024. So this pattern indicates that Shigella flexneri transmission in Riverside County has been largely well controlled. Because that time you can see the 2022 reported case, there's a kind of half a year gap with the 2023 cases. The group 2 and group 3, the group 3 represents about 70% of Shigella sonnei cases reported in 2023 and 2024, indicating an ongoing active transmission during this period. The group 2 includes the earliest cases. You can see the 2020 and 2021 cases, but a few isolates re-emerge in 2023 and 2024, suggesting occasional reintroduction or low-level persistence. So overall, the Riverside counties, based on from this phylogenetic tree, the Riverside counties pattern demonstrate that multiple independent lineages of XDR Shigella are circulated in Riverside County over time with varying levels of recent activities. Next, let's look at the regional patterns across the five counties in Southern California. So here I use the different colored stars to represent cases from different counties. And same as the Riverside County, I use the different color to mark the different cases from different years. As mentioned earlier, the XDR Shigella also cluster into three major groups. First, I will begin with group 1, which contains the same as Riverside County's cases, which contains the Shigella flexneri isolates. So although Riverside County only had 2 cases in this group in 2023 to 2024, however, this lineage is clearly still circulating across other Southern California counties. We have so many cases has been sequenced. You can see many isolates from county A, B, and C. The A, the green one, the county B, the purple one, and C is the blue one. And it forms a large continuous cluster here. So for county D, the 2 of the 4 2024 cases fall within this group. Here's a dark orange case is from county D. So they are separated. They are not gathered closely in this phylogenetic in this group 1. But you can see those 2 cases are not closely clustered with Riverside. Riverside is the light yellow one. Are not closely clustered with Riverside cases. This means their infections are not directly from Riverside County. So overall, the group 1 highlights the active ongoing circulation of Shigella flexneri in the broader metropolitan region in Southern California, even though Riverside itself has only relatively 2 recent cases. The group 2 includes Shigella sonnei isolates and shows a very different pattern with group 1. You can see almost 96% of the isolates in this group are from Riverside County and County A, and barely detect cases from other counties. This suggesting a relatively stable transmission network between the Riverside County and County A during the early period, because lots of the cases in Riverside are 2022. The group 3 is the dominant Shigella sonnei lineage circulating in Southern California during 2023 to 2024. It represents the most actively transmitting lineage in this region. This group contains about 81% of the Shigella sonnei cases reported in 2023 to early 2024. It's same as the Riverside trend. In Riverside, if you can still remember the phylogenetic tree of Riverside County cases, majority of the recent cases are also clustered into the group 3. Within this cluster, we can also see 2 other County D's 2024 cases, and then we can see clearly here that closely grouped with the County A's cases. But not to the Riverside cases. So this suggests that County D's recent Shigella sonnei activity is tied to broad regional networks, but not directly from Riverside County. Let's briefly summarize the results. So the first is the group 1 Shigella flexneri appears largely well controlled within Riverside County during 2023 to 2024. However, it continues to circulate widely in other Southern California counties. Second is the group 3, which are Shigella sonnei, represents the most recent and the most active cluster across the region, including the Riverside. So these cases likely reflect ongoing transmission networks that public health partners should continue monitoring. However, because the whole genome sequence identifies only the genetic relationships but not the behavior connections, so the patient interview records will help to identify the potential epidemiological links between these group 3 cases in Southern California. Third, so based on the available genomic data, County D's cases are not directly linked to those in Riverside County. Maybe potentially just because based on the phylogenetic tree, potentially directly from County A's cases. Here I must mention about the limitations of this research. So first, our historical whole genome sequence data is limited because the state lab did not sequence all Shigella isolates. So they can only sequence about 1/3 of the submitted cases. That means many earlier cases are missing from the data sets I'm analyzed, and therefore they are not appear on the phylogenetic tree. But the good news is our lab was recently PostNet certified and will begin sequencing a greater number of local Shigella isolates. We hope this will significantly improve the completeness of our data sets and allow for more accurate reconstruction of local transmission patterns over time, and will have more more accurate response of public health concern of the XDR Shigella locally. The second is on the phylogenetic tree, the perfect bootstrap value just only means these cases are genetically related, but doesn't mean that direct transmission between the two patients. So even with strong genetic similarity, the missing intermediate cases, for example, due to under-testing or under-reporting, this will still create gaps between those two patients. So therefore, the whole genome sequence provides powerful clues about relatedness, but the epidemiological interviews remain essential to determine the real-world transmission pathways. Together, these limitations highlight the need of ongoing genomic surveillance and complete case investigations of XDR Shigella to provide a more healthy Riverside County and Southern California. Last but not least, I want to thank you our epidemiologist team, and also my lab director and deputy director, deputy health officer for supporting this research. That's the end of my presentation. I'm happy to answer any questions. Thank you. Does anyone has questions?
Hey, yes. This is Curtis. I have a quick question. Yeah, thanks for this presentation. This is really great work, just given the rise of XDR Shigella in recent years. When you were, I forget which slide it was, but you were focusing on a tree of just the Riverside County
Mhm.
cases. I'm curious if you or others, within your health department had epidemiological links between those Riverside County cases. I know it might be hard to get that information for other counties, but I'm just curious if you had a chance to like overlay any kind of epi evidence, whether the folks were in the same MSM communities or
Mm-hmm.
yeah, some other kind of links. I'm just curious if that was considered here. Thank you.
Yeah, thank you, Curtis. The questions are very good. We do have some interviews about the Riverside County because majority of the cases are from a city, two cities. They have large men sex with men population, but we don't have like a connection between other county. Maybe because the XDR Shigella just emerged from like 2020, around 2018 or 2020, I can only get the result whole genome sequencing from the Cedric on 2020. Because previously, I think whole genome sequencing is not very popular um, in public health area. Mm, if we want to link the whole Southern California or even the California area, we need the large collaboration between among counties. So just sorry, we don't have any epic evidence to show the Riverside County results with other counties' results. Yes, that's my answer.
Got it. Thank you.
Thank you.
Yeah, I know at least one other county that might be interested in collaborating if you haven't already talked to them. Yeah, I can follow up with you afterwards.
Okay, thank you so much.
Yeah, yeah.
It looks like there might be one in the chat.
Oh, let me see. The chat, there's a question, if this data was pulled from NARMS, how does the genomic data correlate with the phenotypic in vitro data? Um, I got it from the Cedric, so search for the case number, but it's not the NCBI's SAR number. Then I downloaded each case from the NCBI to get the genomic data. So the part, how does the genomic data correlate with the phenotypic? May I ask, what do you mean the in vitro data?
Oh, hi. Yes, thank you. I believe NARMS pulls the genomic data from the PulseNet databases, and then they perform individual testing for susceptibility, like using MIC concentrations on a typical plate, and then they grow the organism, they test it, and then they read it, and then uh, according to the interpretations of the MIC, they're either sensitive or resistant. So I wanted to learn if there was a correlation between the resistant patterns from the genomic data and then how the results from the phenotypic or MIC testing uh, correlated between each other. If it was resistant on the genomic and if it was resistant on the phenotypic testing. But it looks like you clarified that the data was pulled from Cedric, not necessarily NARMS. And I'm sorry, I think I might have seen on the slide where it said NARMS data.
Mhm. Um, so the I so based on the Cedric, it has been identified XDR Shigella, but I'm not really sure about like how did they do that, like resistant to all those five. Um, I I don't have that information, so I just trust their results. But I will check the norms.
Oh, okay. Okay,
Yes,
thank you.
thank you so much.
Yeah, and the other quick question that I wanted to make is that on the group of the Shigella flexneri resistant isolates, was the serotype determined for the species for flexneri?
You mean the serotype for the XDR Shigella?
Yeah, like sometimes there's like 6 groups in flexneri, but sometimes it's not a routine test. So I was just curious to know if that was determined.
Mm-hmm. So previously, like we send all our PulseNet samples to directly to the state lab. In our lab, we don't do any serology there, but from this year, like we got the certification in October, we just do the sequencing to some
Oh,
of
okay.
them. Yes. Mm-hmm.
Okay. Thank you.
Yeah, thank you. Yeah, Curtis mentioned in the chat there are some bioinformatics tools that can predict Shigella serotypes. Curtis, is it possible you can provide some like what are they or
Yeah,
in Terra or
yeah, sure. So we, at Theogen, we've got some of those tools incorporated into the Theoproc workflows, if you are a Terra user. But otherwise, they are open source tools that are available. Um, the ones I'm thinking off the top of my head are Shigei Finder is a good one. Another one is called Shiga Pass. Um, and please correct me if I'm wrong, but I think PulseNet has incorporated the Shiga Pass tool into their platform, or at least they're planning to in the future. Um, so if you're submitting to the PulseNet, platform and databases, you should get serotypes that way. Again, I'm just uh, uh, stating that off of what I've heard, but yeah, there's another tool from FDA. It's a little bit older. It's also called Shiga Typer. Um, oh yeah, thank you, Karim, for confirming that. But yeah, if since you've got the sequence data, you can at least determine those serotypes and see if there's any further, subdivision of those the flexneri, and I think in group one. The phylogenetic tree is really helpful to cluster the samples together, but it might help to add that layer of serotype just to make sure, because I would assume the serotypes would be identical if there were, um, you know, possible transmission between the patients that were sick with Shigella.
Yeah, thank you, Curtis.
Hmm.
So there's another question. I missed the first three meaning also. So how did you classify group one, two, and three? Yeah. Sorry, I have to mention like I have a meeting in 2 minutes. Sorry about that. So I will answer this last question. If you have any others, just you can email me or like contact me. So the phylogenetic tree, actually, when I pull the data, I already know like which species are Shigella flexneri or Shigella sonnei or Shigella sonnei. So on the phylogenetic tree, I just because all of those Shigella flexneri are clustered together. So I make it kind of like the root or something like that. And then we're can see the Shigella flexneri, all of those cases are grouped together. And the Shigella sonnei, we can see actually if you want to see more in details, we can separate the Shigella sonnei into much more smaller groups, but that will make the explanation more complicated. So I just separated the most two biggest groups as a group two and group three. It's not very critical classification. So I hope I answered your question. Yeah, sorry, my apologies. I have to stop this meeting.
No problem. Thank you so much. That was a very good talk.
Yeah, thank you. But I'm happy to communicate to talk more about it later through email or other opportunities. Yeah, thank you.
Yes, thank you so much. Have a good day, everybody.
Bye.